In this video, I'm going to be explaining the whole of Module 1 Cell Biology for GCSE biology students. And once you've watched the video, if you want to consolidate your learning, you can click this link, fill in your details, and we will send you a full set of flashcards on cell biology immediately for free. Now, if that's not enough, we also have my resources for GCSE biology, and here you'll find complete sets of study notes with full explainer videos, examiner tips and tricks, and extra practice questions. You can click the link to access those too, and again, they are completely free. Yes, you heard it right. Now, let's get going with the video. Let's start with animal cell structure. So animal cells are eukaryotic cells. This basically means that they have a nucleus and other membrane-bound organelles, such as mitochondria. So we're going to go through the key subcellular structures or the key organelles. First of all, the nucleus, which you can see here on the diagram. Now, the nucleus contains DNA, which will be arranged in chromosomes, and we say the nucleus controls the cell's activities. So two things to remember there. Then we have the cytoplasm. The cytoplasm is a jelly-like fluid that fills the cell, and it's where most chemical reactions occur. So you'll find lots of enzymes in the cytoplasm that control chemical reactions. Next up, we have the cell membrane. Now, the cell membrane controls the movement of substances in and out of the cell. The word controls is important here. Don't just say it lets things in and out. We say it controls the movement of substances in and out of the cell. And it is partially permeable. This means selected substances can pass through and other substances cannot. So, for example, glucose and oxygen, which are both needed for respiration, they are able to pass through the cell surface membrane and into the cell. Next, we have the mitochondria, another membrane-bound organelle, same as the nucleus. Now, the mitochondria are the site of aerobic respiration, respiration that uses oxygen. And here, with respiration, it releases energy that the cell needs for various processes. Finally, we have the ribosomes, which are a really small subcellular structure. You can see they're just dots. They are the site of protein synthesis. So this is where proteins are made from amino acids, and they are found in the cytoplasm. Let's move on to plant cell structure. Now, plant cells are also eukaryotic, which means they also have a nucleus, and they do have some extra subcellular structures or extra organelles compared to an animal cell. So let's go through those structures. For example, unlike an animal cell, plant cells have a cell wall around the outside of the cell. So the cell wall is outside of the cell membrane. The cell wall is made of cellulose, which is important to remember. It's a rigid structure. It supports the cell. It protects the cell. It's fully permeable to water and dissolved substances, so substances can pass through the cell wall. Remember, it's the cell membrane that we describe as partially permeable. Plant cells also have a large permanent vacuole, which you can see here, this big blue section. Now, this vacuole contains cell sap, which is a weak solution. It will contain sugars, salts, and water. It helps to maintain the internal pressure when it's nice and full, and it pushes out against the cell membrane, and it helps to support the plants. Plant cells can also have chloroplasts, unless we're talking about a root cell, which is underground, so it does not. But plant cells above the ground will have chloroplasts. Now, these chloroplasts contain the green pigment chlorophyll, and chlorophyll's job is to absorb light energy for photosynthesis. So we say the job of the chloroplasts is photosynthesis, where plant cells make their own food, which is obviously glucose or sugar. Remember, chloroplasts are not found in root cells. They are underground. They do not receive light energy from the sun. They cannot photosynthesize. As well as these extra 3 structures, they also have the same subcellular structures that we went through with the animal cell. So they also have a nucleus, they also have cytoplasm, they will have a partially permeable cell membrane, which remember is inside the cell wall. So the cell wall is on the outside, which you can see in green. The cell membrane is inside of that, which you can see in brown. They also have mitochondria for aerobic respiration, and they will also have ribosomes for protein synthesis. Same functions as animal cells, so I'm not going to go through them again, but you should be able to recall all of the functions of these key subcellular structures. Let's have a look at a prokaryotic cell. Now we're talking about bacterial cells here. Bacterial cells are prokaryotic, not eukaryotic. They are about 10 times smaller on average than a eukaryotic cell. They are simpler than eukaryotic cells, and we call them prokaryotic cells mainly because they do not have a nucleus and they do not have any membrane-bound organelles. So no nucleus, no mitochondria would be another example of an organelle that they do not have. So what do they have? They do have a cell wall, which you can see here. This cell wall, if you look closely, it's the layer that's highlighted in blue, I think. Yeah, the blue layer. There is a green layer around the outside, which we'll get to in a second. Now, the cell wall, just like in a plant cell, it does provide structural support and protection, but it is not made of cellulose. It's actually made of a different molecule called murin. Now, you don't have to remember the name of that molecule, but you should be able to give as a difference between, say, a bacterial cell and a plant cell. They both have cell walls, but the cell wall of a bacterial cell is not made of cellulose, whereas the cell wall of a plant cell is made of cellulose. They have a cell surface membrane just the same as the cell surface membrane in an animal cell or a plant cell or eukaryotic cells. So same job, it is partially permeable. It does control what enters and leaves the cell. And just like with our plant cell, the cell surface membrane will be inside of the cell wall. So if the cell wall is this blue layer, the cell membrane is just sitting inside of that cell wall. It also has cytoplasm, which you can see in yellow. Again, just the same as in a eukaryotic cell. It's a jelly-like fluid that fills the cell. It contains enzymes because it's where lots of different chemical reactions take place. But in this cytoplasm, there are no membrane-bound organelles. So there's going to be no nucleus and no mitochondria. The DNA then, because there is no nucleus, the DNA is just loose within the cytoplasm. And it's not linear, so it's not packaged into chromosomes, it's actually circular. So you can see you've got a kind of big circular, looks a bit messy, doesn't it? But that will be a big circular loop of DNA in the cytoplasm, not enclosed in a nucleus, which is why these are prokaryotic cells. But just like a eukaryotic cell DNA, it still carries genetic information. It's what we describe as the genetic material of the cell. Now, just like a eukaryotic cell, prokaryotic cells also have ribosomes, and again, they are the site of protein synthesis. However, it might be worth remembering, if you are asked for a difference between a prokaryotic and a eukaryotic cell, the ribosomes in a prokaryotic cell are smaller than the ribosomes in a eukaryotic cell. So they're smaller than the ribosomes in a plant cell or an animal cell, but they have the same job, making proteins. Let's have a look at some of the structures that could be found in a prokaryotic cell, but are not always present. So most prokaryotic cells have plasmids. You can see a plasmid here. There's only one in this bacterial cell, but there might be multiple plasmids. And these are smaller loops of DNA, so small circular DNA molecules, and they can be exchanged between bacterial cells, so they can transmit genetic material from one bacterial cell to another. Some bacterial cells have a slime capsule, which is this green layer that we mentioned earlier. This is the very outer layer of the prokaryotic cell. It's not always present, but if it is, it's a slimy outer layer that sits just outside of the cell wall, and it helps to protect the bacterium. It stops the bacterium from drying out, you could say. It also helps the bacterium to adhere to or stick to surfaces, and also helps bacteria to stick to each other as well. This bacterium has a flagellum, a tail-like structure. Again, not always present. Some bacterium just have one flagella, like here, there's just one flagellum, but some bacteria have multiple flagella, which is the plural. This is a tail-like structure that's used for movement. And there is an animal cell that has a flagellum, and that would be a sperm cell, which obviously helps the sperm cell to swim towards, move towards the egg cell for fertilization. Now let's move on to thinking about the light microscope. You will have to use a light microscope during one of the required practicals in Module 1. So if we start by looking at the layout of this type of microscope, it's called a light microscope because it uses light to help you see your specimen. So there'll be a light just underneath the stage, and the light is going to shine up through a little hole in the stage, and on that stage you're going to place your slide, which will have your specimen on it. Obviously, we can describe this part of the microscope as the base, but what's important here are the lenses and the adjustment wheels or the adjustment knobs. So there are two lenses to consider. The eyepiece lens here at the top, that's the one that you're going to look down, so you're going to put your eye here. And then you've got your objective lenses here. Now, light microscopes will have multiple objective lenses, maybe 3 or 4. Each of those objective lenses will have a different magnification or a different power. So a higher power, a higher magnification, lower power, lower magnification. On the side of the microscope, and we can call this part of the microscope the body, or you might see it referred to as the arm, doesn't really matter, you're not going to be asked about that. But on that part of the microscope, you'll see some adjustment wheels or adjustment knobs. You've got the coarse adjustment knob to get your specimen into rough focus, and then you can use your fine adjustment knob, which is usually a little bit smaller, to get it into perfect or really clear focus. So why do we use a light microscope? Well, cells are really small. They cannot be seen with the naked eye. So microscopes are going to help us to see that cell because it's going to produce an image of the cell that is larger, bigger than the actual cell. Now we need to have two key definitions. The magnification is how many times larger the image is compared to the actual size. So we're not going to say how zoomed in it is. We're going to say the magnification is how many times larger the image is compared to the actual size. Another key definition is resolution. Now, obviously, the higher the resolution, the clearer the image or the more clarity the image will have. But the definition of resolution is actually this. It's how easy it is to distinguish between 2 points that are close together. So for example, if you had 2 points, 2 organelles, maybe 2 mitochondria, if they are very close together and your microscope has a low resolution, you would not be able to distinguish them as 2 separate objects. And they would look like one blob. But with a higher resolution, you are able to distinguish between those 2 points or those 2 objects that are close together. So this does lead to better clarity, a clearer or a more detailed image, the higher the resolution is. Light microscopes have quite a low resolution, especially compared to an electron microscope. So really small organelles like ribosomes would not be visible. You would not be able to distinguish those ribosomes as separate objects. Let's think about our required practical. So during your required practical in Module 1, using the microscope, you would have to prepare a slide to view under the microscope. You're probably going to look at onion cells. So let's go through that example. You do need to peel a thin layer from an onion, and onion cells are obviously plant cells. You would take that thin layer or that layer of tissue and place it, making sure it's nice and flat, on a glass microscope slide. You're going to need to add a stain. So we're going to add a drop of iodine solution, which has an orangey colour. That's going to stain the cells and it's going to stain the subcellular structures to make them visible. So you might be able to see the nucleus, you might be able to see the cell wall, possibly the cell membrane, not much else. Remember, ribosomes are too small. Mitochondria, the light microscopes that you use in school probably do not have a high enough magnification or a high enough resolution to even see the mitochondria. So you're probably just going to see the nucleus, the cell wall, the cell membrane. You can use a mounted needle, which is kind of a wooden handled instrument with a long metal needle part. You can use that mounted needle to carefully lower a cover slip over your sample, and we lower it slowly and we lower it at an angle to avoid any air bubbles that would make it quite difficult for you to see the cells. Now, key things to remember: the specimen needs to be thin. Ideally, we want it to be a single layer of cells, and this is so the light, which is shining down from here, can pass through the tissue so you can actually see the cells and any subcellular structures that may be visible. We also do need to use that stain, that iodine, which is orange. It helps to provide contrast. And again, it's about making the subcellular structures more visible, because they're not going to be visible. They're going to be colourless without that stain. So making sure it's thin so light can pass through, using a stain to increase the contrast. Both of these things are going to help us to see the subcellular structures. Now, just a point before we move on, remember these are onion cells. Onions grow underground. There will be no chloroplasts in these cells. They do not photosynthesize. Let's have a little look at a diagram which would show us our prepared slide. So the glass slide is the rectangular shape. You can see your specimen placed on the slide, which has been stained with iodine solution. You might have added a little bit of water as well to your slide, just to help your onion cells to stick. And then the cover slip is the smaller, thinner glass square that you place over the top of your specimen. It helps to make sure it's nice and flat. It squashes it into a single layer of cells. But make sure you do lower the cover slip slowly, using a mounted needle at an angle to try and reduce the number of air bubbles. Let's think about risk assessment could come up. Now, the way we do risk assessments for practicals in science is we identify the risk, we identify the hazard, which is what could happen, and we identify a control measure. And the mistake that students make in exams is their risk assessments are far too vague. They will just say things like, wear a lab coat, wear goggles. You need to pick risks and give control measures that are specific to your practical. So for example, in this practical, we might be using a scalpel to cut the onion. Now, obviously, scalpel blades are sharp. You could cut yourself. So when you cut, you always cut with the blade facing away from your fingers, and you should be cutting on a hard surface, such as a white cutting tile or a dissection board. You also should use a sharp scalpel. I know that sounds kind of contradictory, but sharp scalpels are safer than blunt scalpels, because if it's sharp, you can make one clean cut. You're less likely to have to move the scalpel back and forth, which is actually more risky in terms of cutting yourself. Using the mounted needle is another risk. Now, you're going to be using the mounted needle to maybe flatten the piece of onion tissue and to lower the cover slip, but they are sharp, so you could cause injury. Again, you could cut yourself, poke yourself. So always point the sharp end away from your hands and away from any other people. The iodine solution, now iodine solution is an irritant. I've put that in bold because that's a really good word to use. It's an irritant. It can irritate your skin. It can irritate your eyes. So for this one, we can say wear safety goggles, but to make sure it's specific, we say so iodine does not enter the eyes because it's an irritant. And if you do spill any iodine on your skin or even just on your desk, you should wash it with water immediately to dilute that iodine, or put water onto the spill on the desk and then clean it up with paper towels so that no one else touches the iodine. If you do spill anything, obviously if the floor becomes wet, you could slip. So dropped slides, water, iodine could make the bench or floor slippery. You could potentially fall over and hurt yourself. So wipe up spills straight away and keep your work area tidy and dry. Hopefully you can see that all my control measures are very specific to these specific risks for this practical. Let's continue thinking about how we would actually use the microscope. So we've already prepared our slide, we've already done our risk assessment. Now we want to view our specimen, our onion cells. We are going to start with the lowest power objective lens, okay? So the lowest power objective lens. It will probably be the shortest objective lens. That objective lens has the lowest magnification. And we use the lowest power objective lens first, so we get a nice wide field of view, and then we can decide which specific area we want to magnify even further. You can use the coarse adjustment wheel or the coarse focus wheel or the coarse focus knob, whatever you want to call it, to bring the slide into general focus. Then use your fine focus knob or fine adjustment knob to sharpen the image to get it clearly in focus so you can see the cells and any subcellular structures. Once you've got it in as good a focus as you can switch to a higher power objective lens, okay? So you're going to be turning this here to turn the objective lenses, and you can now switch to a higher power or a higher magnification objective lens, and you might have to slightly refocus, hopefully just using the fine adjustment knob now because it should be roughly in focus anyway. If you're asked to calculate the total magnification, which you might be, there are two lenses to consider because the eyepiece lens has a magnification and the objective lens that you've been using has a magnification. So the total magnification is calculated by multiplying those two together. So the eyepiece lens magnification times the objective lens magnification gives you the total magnification. For example, if the eyepiece lens had a magnification of ×5 and the objective lens had a magnification of ×40, the total magnification of your specimen using those lenses would have been 200. It's 200 times larger than the actual specimen. You might have to do a biological drawing with this practical. So here is an image that was seen under a light microscope, and you can see the magnification, the total magnification used was ×200. You can see the cell walls, you can see the nuclei, they've been stained orange with iodine, you can't see much else. And here next to it, we have a very good biological drawing. So let's go through the rules for doing a biological drawing. It has to represent what you actually see under the microscope. You should be using a sharp pencil and only drawing what you see. So don't make up and draw some mitochondria or some ribosomes. If you can't see them, don't draw them. Use continuous single lines. You can see that on this drawing here. They are continuous. There's no breaks in the lines. They are nice, clear and crisp, continuous lines. Label the structures you can see. You can see on the biological drawing here, we've used ruled label lines and we've only labelled the structures that are visible that we can identify. There should be no shading, no colouring on a biological drawing, and you should try to include a title, for example, onion cells under the light microscope, and the magnification that was used. So on this biological drawing here, in one of the corners, you would add the magnification. So the person looking at your biological drawing knows how much bigger that drawing is compared to the actual size of those cells. Now you'll notice that we've only drawn 3 cells, not every single cell. And in your exam, they would probably instruct you, draw 3 of the cells or draw 2 of the cells. But they're probably not going to make you draw every single cell because that would take you far too long in your exam. So make sure that you follow the instructions in the question. And if you are asked to improve on an image, remember the key rules so you can suggest possible ways that the person might have done a better biological drawing. From my experience, usually the magnification is missing, so you can suggest adding on the magnification to the biological drawing. Let's think about some specialised cells. We're going to go through some examples of specialised animal cells and some examples of specialised plant cells. Now, specialised cells are just cells that have adaptations that help them to perform a specific function or a specific job effectively. And specialised cells arise through a process called differentiation. Differentiation is the process by which a cell becomes specialised. So let's look at 4 key examples of specialised animal cells. The first image we've got here is a sperm cell. Now you might be asked to describe its function. So sperm cells are obviously produced by males. They're produced in the testes. They are a gamete or a sex cell. Their function is to swim towards the egg cell for fertilisation, where one sperm cell will fuse with the egg cell. They've got adaptations to help them perform this function. So they do have a flagellum, which other animal cells don't have. Remember, they're more commonly found in prokaryotic cells, bacterial cells, but the sperm cell is the exception. So the flagellum obviously is used for movement. So that tail is going to help it swim towards the egg. They will have lots of mitochondria. So in this middle region here, there'll be many mitochondria. Remember, mitochondria are the site of aerobic respiration. So more energy is going to be released, and that energy can be used for movement or to help the sperm cell actually swim towards the egg cell. Other adaptations that the sperm cell has, there's an acrosome. Now you don't need to remember that key word, but basically if you look at the head here, the tip of the head, you've got this region. You can call it an acrosome if you want to, don't have to, but in that region there are enzymes that will break down the egg cell membrane, and the egg cell will only allow one sperm through, so only one sperm cell nucleus will enter the egg cell, but those enzymes help when the sperm cell reaches the egg to break down the egg cell membrane. And something else we should know, in this head region here where we find the nucleus is haploid. I always think H for haploid, H for half. It has half the number of chromosomes of a normal human body cell. So in our body cells, they have 46 chromosomes, but in the sperm cell, it will only have 23. And this is obviously because it's going to fuse with an egg cell. The egg cell will also have 23 chromosomes. So when they fuse at fertilization, you will have the full number of chromosomes, which we call the diploid number. The next cell we're going to look at is this one here. This is a nerve cell, also known as a neuron. Now, the function of the neuron is to carry or transmit electrical impulses around the body. Make sure you say electrical impulses and not messages or information. Now, how are they adapted to do that? They have this long axon. See this portion here? This long axon to allow the electrical impulses or the electrical signals to travel long distances. They're also branched. The branches are called dendrites, but you can just call them branched ends. That allows each neuron to connect with other neurons so that the electrical impulses can be transmitted from neuron to neuron. They also have a myelin sheath, which is a fatty layer of cells that wraps around the axon. We can just say the myelin sheath insulates the axon, a bit like how plastic insulates the wires in your home. And it also speeds up the transmission, so the electrical impulses travel more quickly. Let's look at two more. The next one we're going to look at, which is quite a common one to come up, is a red blood cell. Now, the function of a red blood cell is to carry or transport oxygen. So they pick that oxygen up in the lungs and they will carry that oxygen in your bloodstream around your body to all your cells or tissues. So in terms of adaptations, they have a biconcave shape. What does that mean? Well, it's got two concave sides. Bi means two, concave is this kind of sunken in shape, right? So two concave sides or biconcave shape. That is to increase the surface area for gas exchange, because obviously oxygen needs to diffuse in, so the red blood cells can carry the oxygen to the cells, but it will then also need to diffuse out to deliver it to the body cells and tissues. So having that biconcave shape gives it a larger surface area for faster diffusion. They don't have a nucleus. So this is an animal cell with no nucleus. That means there's more room for hemoglobin. Now, hemoglobin is a protein found in red blood cells, and it's actually the hemoglobin that carries the oxygen. When hemoglobin is carrying oxygen, we call it oxyhemoglobin. So without having a nucleus, there's a lot more space for much more hemoglobin, so they can carry more oxygen. And as we've said, they contain hemoglobin. That's another adaptation, right, to carry the oxygen. They are very small and flexible, so they can fit through the narrowest of blood vessels, which would be the capillaries. In fact, the capillary diameter is about the same as one red blood cell. So they squeeze through the capillaries, one red blood cell at a time. The final animal cell we're going to look at is this one here, which is a muscle cell, also known as a muscle fibre. Their function is to contract and relax. So they shorten and lengthen, contract, shorten, relax, lengthen. And that's to allow movement in the body. So in terms of adaptations to help them do their job, they are long and thin, so they can contract efficiently. They have many mitochondria. I think that's quite an obvious one. Many mitochondria. Mitochondria are the site of aerobic respiration, so they can be provided with lots of energy because muscle contraction requires a lot of energy, which will be released from respiration. They can store glycogen. Glycogen is a store of glucose, so it's many glucose monomers joined together. And what muscle cells can do is break down the glycogen into glucose, so they've always got glucose to use for respiration, so they can always release energy for contraction. They also have special protein filaments. There's lots of protein filaments in a muscle fibre. And these protein filaments slide over each other, and that's what allows the muscle fibre to change length, to shorten when they contract and to lengthen when they relax. We're going to look at some specialised plant cells now. The first one we're going to do is a root hair cell. So obviously these are underground, nothing to do with your hair. They are plant cells. They are found on plant roots. So the job of a root hair cell is to absorb water from the soil and mineral ions from the soil, such as nitrate ions, magnesium ions, phosphorus ions, that kind of thing. So in terms of adaptations, they have what we describe as a root hair. Now this is a long extension. You can see the cytoplasm and the cell membrane and the cell wall extends in this portion called the root hair. That's obviously, again, about increasing the surface area, because the bigger the surface area, the more absorption, whether that be of water or mineral ions, can take place. They do not contain chloroplasts. It would be a waste of energy for them to make the chloroplasts. They're underground, there is no light, and therefore there will be no photosynthesis. They will have many mitochondria, because they do require a lot of energy from respiration. Now, we'll be talking about active transport later in this video, but root hair cells can absorb mineral ions by active transport. Active transport does require energy because you'll be moving those mineral ions against their concentration gradient. So because it's an energy requiring process, they will have many mitochondria to release sufficient energy. Let's look at a palisade cell. This kind of looks like a typical leaf cell, right? But this is a palisade cell, so it does have some adaptations. To give it its full name, it's a palisade mesophyll cell. That just means it's a cell that's found in the leaf, and specifically, it's a palisade cell. The function is to carry out photosynthesis. So this is where most photosynthesis takes place in a leaf. This is the cell type that is primarily responsible for photosynthesis. So think about the adaptations. Well, it's going to be packed with chloroplasts. Remember, chloroplasts contain chlorophyll, so they can absorb light energy for photosynthesis. It's all about maximizing the rate of photosynthesis in these cells. They're going to be located very near to the top of the leaf. Just above them, you have what we call the upper epidermis, but they are right below that. So near the top of the leaf, again, this is to absorb maximum sunlight for photosynthesis. You'll notice they're rectangular shaped. Now this is great because it means they can tightly pack together. So you have many palisade cells next to each other, packed tightly together near the top of the leaf. So you've got as much surface area for light absorption and as many chloroplasts for light absorption as possible near to the top of the leaf. Two more specialized plant cells. So we're going to look at xylem cells. Now the function of xylem cells is to transport water and minerals up the plant from the roots, to the leaves. Also, the xylem vessels do provide some support, okay? So this is one of the cells that students probably find hardest, I would say. But if you look at the diagram, the xylem cells, I can actually see 1, 2, 3, 4 xylem cells here. They kind of stack on top of each other to form this tube or this vessel, xylem vessel. And you can imagine, can't you, that within those cells or within that xylem vessel, you've got water moving up. It's always going to go up from the roots to the leaves. And as well as water, you will have mineral ions being transported. Let's go through the adaptations. So the cells themselves have no end cell walls. So if you look at each of these cells, you would expect there to be an end cell wall, but there are no end cell walls, which helps these cells to form a continuous and hollow tube so that the water can flow easily between the cells. The cell walls are strengthened with a substance called lignin. Now, lignin is really strong, so it does help with structural support. We said that here, didn't we? It helps to support the stem, and it also waterproofs the xylem so that water does not leak out of the xylem vessels as it is being transported up. In these cells, there is no cytoplasm. There's none of that jelly-like cytoplasm. There is no nucleus. And again, this allows the water to flow more easily, or you could say it means it does not restrict the flow of water. Now, because these cells have no end cell walls, no nucleus, no cytoplasm, they are actually dead cells. Hollow dead cells that form this tube to transport water and mineral ions. Now, the transport of water and mineral ions, it comes up again in a later topic, but you might know that it is referred to as the transpiration stream. So you could say that the xylem cells are involved in the transpiration stream, which is just the transport of water from the roots up to the leaves. Let's have a look at our last plant cell. These are phloem cells. So phloem cells form phloem vessels, just like xylem cells form xylem vessels. And the phloem vessels will be next to the xylem vessels, but they do not transport water and mineral ions. In fact, their function, it is transport, but they transport sugars, mainly the sugar sucrose. Also, some amino acids will be transported in the phloem, which you can see on the diagram. But for GCSE, you can just say the job of the phloem is to transport sugar or sucrose. And this process is known as translocation. But different to the transpiration stream, the phloem can transport sugars both up and down the plant. So the sugars can be transported in both directions in the phloem. So, just like the xylem cells, the cells that form the phloem vessels do not have a nucleus. Again, this is about making sure that the flow of sugar is not restricted. They have some cytoplasm, but very little cytoplasm, and it will be pushed to the edges of the cell. So that's going to help the sugar to flow or move in the phloem cells more easily without being restricted. The end walls, they do have end walls, unlike the xylem cells, but the end walls are perforated. They have little holes in them. We actually refer to them as sieve plates. And because the sieve plates have little holes or little pores in them, it allows the sugar to move more easily from phloem cell to phloem cell. Next to the phloem cells, you can see one here, you have what we call a companion cell. So they sit next to the phloem cells and they contain many mitochondria to actually provide energy for the phloem cells themselves. Okay, let's continue with different types of microscope. Now, before we put any text, you should be able to see from the images, we're going to be looking at the light microscope, which you use in your own required practical, but we're also going to be looking at the electron microscope and comparing the two. So we know that microscopes are used to view cells and cell structures, organelles, because they're too small to see with the naked eye. We've got both the light and the electron microscope. So the light microscope, or you may see it called the optical microscope, but its key features, it uses light and it focuses the light using lenses, glass lenses. And the glass lenses will help to magnify or make the specimen bigger than it is in reality. Now, with a light microscope, you can view living specimens or dead specimens. The magnification is lower than an electron microscope. You don't have to know the number. I've given you the number here, up to maybe 1,500 times bigger. But you just need to know it's lower than the electron microscope. The resolution is also lower. Remember, the resolution is the ability to distinguish between two objects as separate objects. The resolution is going to be lower than the electron microscope. You'll notice it's given as a distance because that is the distance at which you can still distinguish objects as separate objects. But if they're any closer together than that distance, you will not be able to distinguish them as two separate objects. You can get colour images, so you'll be able to see any colours of cells or any colours of stains that you may have used. And you're going to use a light microscope for viewing whole cells, tissues. You probably looked at onion tissue. You might even view a living organism, a fly, something like that, under a mite, Daphnia. I'm trying to think of what you might have seen, because you can see living specimens with a light microscope. Now, electron microscopes does not use light. They use electrons. And because you're using electrons, the specimen has to be placed in a vacuum where there is no air. So you can only view dead specimens, not living specimens. They have a much higher resolution. So two structures can be incredibly close together and you can still view them as two separate structures. They have a much higher magnification. And because the magnification and because the resolution is so much higher than a light microscope, you can get detailed images of not only cells, but internal structures. You can see the detail of the organelles, even the smallest of organelles can be seen. However, you can only get black and white images because you're using electrons and not light. And it does require specialist skills to use an electron microscope. The preparation process is complex. For example, the specimen has to be incredibly thin and a complex staining process is used. So it will be very time consuming and you will need to be a specialist scientist to use one of these microscopes. These are going to be used if you actually want to see the subcellular structures, so the organelles inside the cell, like the mitochondria or the ribosomes, or possibly you want to look at a very small specimen like a virus, which is even smaller than a bacterial cell. In this slide, I've got a nice comparison for you. So this is the level of detail that you need in case you have to do a compare question, give the differences style of question. So the light microscope uses light, the electron microscope uses electrons. In terms of focusing them as well, I might just add that on. The light microscope uses light, which is focused using lenses. The electron microscope uses electrons, which is focused using magnets. Now, the resolution is much higher in an electron microscope. The magnification is much higher in an electron microscope. But with the electron microscope, you can only view dead specimens. With a light microscope, you can view living things. With a light microscope, one of the benefits is you can see colour. So if you've stained it, you'll see the colour of the stain. With the electron microscope, you only get black and white images. The light microscopes are lower cost, portable, less of a preparation time and fewer skills are needed. The electron microscope is far more expensive, it's much larger, and remember you have a much more complex, time-consuming preparation process. In terms of exam questions, I would not talk about cost in the exam because it's often not on the mark scheme, but any of these features would be acceptable as differences between those two types of microscope. The next thing to look at, you may be asked in your exam to calculate either the magnification, if you're provided with the image of, say, a cell, or you might be asked to calculate the actual, like the real life size of the cell. Now, the formula for magnification is image size divided by actual size. All units must be the same before calculating, so always check that. You may need to convert units, for example, from millimeters to micrometers. You may be asked to give your answer in standard form, so always read the questions carefully. You might need to rearrange the equation because it might not be the magnification that they're asking you for. But this is the formula triangle that you can use if you need help with rearranging equations. You can jot it down on your exam paper. I call it the I am triangle to help me remember where to put the letters. So the way this triangle works, if you're calculating magnification, you can see that the sum you need to do is the image size divided by the actual size. However, if you're asked to calculate the actual size of a cell, you could circle that one, and the calculation you would then need to do is the image size divided by the magnification. Now, questions do often include unit conversions, so I'm going to go through how you would do that. This diagram I find really helpful. So if you're going from meters to millimeters, you times by 1,000 because there are 1,000 millimeters in a meter. Millimeters to micrometers, which is probably the most common conversion on a biology exam, you times by 1,000 because there are 1,000 micrometers in 1 millimeter. If you have to go all the way to nanometers, there are 1,000 nanometers in a micrometer. So again, you would times by 1,000. If you had to go from millimeters all the way to nanometers, you could do it in 2 steps, so times by 1,000 and times by 1,000, or just add those conversions together and times by 1,000,000. Obviously, if you're going back the other way, it would be divide by 1,000. Now, in biology, we don't often use centimeters, but if you do see a centimeter, then you should know that 1 centimeter equals 10 millimeters, or 1 centimeter contains 10 millimeters. So if you're going from centimeter to millimeter, you can see here, you'd have to times by 10. If you're going back the other way from millimeter to centimeter, which is unlikely, but you would have to divide by 10. Let's have a look at an example question. So it says here, an image of a bacterium measures 2.5 centimeters. So that's the image size. They're telling you the image size, or you may have had to measure that image size, but we know the image size is 2.5 centimeters. When it was viewed at a magnification of times 500. So we've got the image size and we've got the magnification provided. Calculate the real size. So they're asking you to calculate the actual size of the bacterium. And it does say give your answer in micrometers. So the first thing I would do is use my formula triangle and think, well, I want the actual size, which I've already got circled. So I'm going to be doing the image size divided by the magnification. And I would probably write that out, because it might be that I get a mark for writing out the sum that I'm going to do. So the actual size equals the image size divided by the magnification. Write it out. Don't just sketch the triangle into your exam paper and expect to get the marks. Actually write it out in words. So the actual size equals the image size divided by the magnification. That's the sum that I need to do. However, they want my answer in micrometers, and I can see that they've given me the image size in centimeters. So I'm going to convert that straight away into micrometers. I could do it at the end, but I might forget. So it's probably best to do it first. So first of all, I'm going to convert centimeters into millimeters. Now there are 10 millimeters in 1 centimeter. So I'm going to times by 10. To convert that into millimeters. But I still need to go ahead and convert millimeters into micrometers. Now there are 1,000 micrometers in a millimeter. And you can see on the diagram here, I'm going to be timesing by 1,000. Okay, so now I've converted the image size into micrometers. They want my answer in micrometers, so I'm done with my conversions now. Do it first so you don't forget to do it at the end. So now I've got my image size in micrometers. I can go ahead and do image size divided by the magnification. Let me get my calculator. Image size divided by magnification. So 25,000 micrometers was the image size. The magnification was 5,000. So my answer for my real size is going to be 5. And I've already converted. So 5 micrometers. The next thing in Module 1 is to learn about mitosis and the cell cycle. Now, cell division by mitosis is important for growth and development of multicellular organisms, or organisms that are made of many cells. In fact, it is used for growth, replacing cells, repairing tissues, and in some instances, some organisms can use mitosis for reproduction. This would be a type of asexual reproduction that comes up when we study topics in paper 2, okay? So make sure you learn the uses of mitosis: growth, replacing cells that have been damaged, repairing a tissue that has damaged cells, and in some instances, it can be used for reproduction if the organism uses asexual reproduction. Now, cells are in what we call the cell cycle, which is a series of stages that a cell will go through as the cell grows and divides. Now, the main stages of the cell cycle. Stage 1 is the longest stage. This stage is actually called interphase. And you can see the purple is interphase on this diagram. So it's the longest stage of the cell cycle, or the stage that a cell spends the longest amount of time in. Now, you don't have to learn the word interphase for GCSE, but you should be able to tell the examiner some of the things that happen during stage 1 or interphase. So the cell will grow larger. The DNA will be replicated, so each chromosome is copied. And subcellular structures or organelles, such as mitochondria and ribosomes, will increase in number or be replicated or duplicated. This is the stage when a cell is preparing to divide. It's getting ready to divide into two. The second stage, which you can see in light blue, is mitosis itself. Now, mitosis is where the nucleus divides to form two genetically identical nuclei. We actually call mitosis nuclear division. So by the end of mitosis, you will have two genetically identical nuclei. You do need to know the name of this stage. You do need to refer to it as mitosis, and we're going to talk more about mitosis on the next slide. But after mitosis, so after you've got your two genetically identical nuclei, we then go into stage 3, which you can see is the green portion on my cell cycle. It's the last stage, the final stage of the cell cycle, the final stage of cell division. It's called cytokinesis. Again, you don't need to call it cytokinesis at GCSE, but you can do because it can get you a mark. What you do have to know, though, is what happens during that third and final stage. So this is where the cytoplasm divides, and we will end up with two genetically identical cells. We say that we make two daughter cells. That's what we call them. So let's look specifically at mitosis. Now, don't forget, before mitosis, you had that first phase, which you can call interphase, where we said the cell does grow, it does increase the number of subcellular structures such as ribosomes and mitochondria, and really importantly, before mitosis, the DNA will replicate, so you will have two copies of each chromosome. Once that's happened during interphase, we can then go into mitosis. So the chromosomes will condense or thicken, and they will now be visible. The nuclear membrane breaks down. The chromosomes will line up in the middle of the cell, or we can say they line up on the equator, if you want to. And one set of chromosomes will be pulled to each opposite end of the cell, and then new nuclear membranes form around each set of chromosomes. So it will look a little bit like this. The chromosomes will line up in the center of the cell, or the chromosomes will line up on the equator. They will be attached to proteins that we call spindle fibers, like this. And what will happen is those spindle fibers will shorten. And if you look at these chromosomes, they will separate down the middle. So one set of chromosomes will be pulled to this end of the cell, and the other set of chromosomes will be pulled to the opposite end of the cell. Okay. Now that's going into slightly more detail than you need for AQA GCSE biology, but it is really important that you can use this phrase here. So during mitosis, one set of chromosomes is pulled to each opposite end of the cell, and new nuclear membranes form. So at the end of mitosis, you will end up with two nuclei. And then we go to stage 3, which you can call cytokinesis. This comes after mitosis. The cytoplasm will divide, the cell membranes will divide, and you will now have 2 genetically identical cells, each with their own full set of chromosomes. Because don't forget, before this took place, all the chromosomes were replicated so that each daughter cell will have a full set of chromosomes, and they will be genetically identical. Now, you may have heard of the names of, prophase, the names of the stages of mitosis, but this really is A-level biology. You do not have to know the names of the different stages of mitosis. You do not need to be able to describe those stages in significant detail. Just saying one set of chromosomes is pulled to each opposite end of the cell, and then new nuclear membranes form is enough. And then after mitosis, the cytoplasm and the cell membranes divide. Let's look at a summary then. So with mitosis, we are going from 1 parent cell to 2 daughter cells. So there's just 1 division involved. You will produce 2 daughter cells, as you can see here. The daughter cells will be diploid, so they will each have a full set of chromosomes, or the full number of chromosomes, because remember, the chromosomes were replicated prior to mitosis, and then 1 set of chromosomes was pulled to each opposite end. The daughter cells will be genetically identical, so they will have the same chromosomes with the same genes. They'll be genetically identical to each other and to the parent cell. And remember, the purpose of mitosis is for growth, so an organism can get bigger, repair of tissues, replacing cells if they've been damaged, and some organisms use it for asexual reproduction. Let's talk about binary fission. We don't have to talk a lot on this, but binary fission is the key scientific term given to the process by which prokaryotic or bacterial cells without a nucleus, how they divide. They don't have a nucleus, right? So they cannot divide by mitosis. So it's a similar process, but there are key differences, and we call it binary fission. The DNA will be replicated just like before mitosis, we replicate the DNA. The prokaryotic DNA is also replicated. So they will replicate their main circular DNA. They will also replicate any plasmids that they have. And then the cytoplasm and the cell membranes will divide. But the main thing for you to remember is in prokaryotic cells, cell division takes place through a process called binary fission. Now, you might be asked to calculate the number of bacterial cells that would be present after a certain number of divisions. So the formula you would use for that would be this one here. So the number 1 represents the starting population of bacteria. So I've assumed here that we've just started with 1 bacterium, but you might be starting with 10 bacterium, in which case that first number would be a 10. So that's where you put the starting number, which is probably 1, and you multiply it by 2 to the power of n, where n, it says it here, where n equals the number of divisions. And the reason we use this formula is every time the bacterial cell divides, it's doubling. It's going from 1 to 2, 2 to 4, 4 to 8, 8 to 16. So we're doubling the number each time. So for example, if you start with 1 bacterial cell and the examiner wants to know how many bacterial cells would be present after 8 divisions, you do 1, because your starting population is just 1 bacterium, and you'd multiply it by 2 to the power of 8. So in theory, you can just do 2 to the power of 8 here, right? You don't really need to include the 1. So what that's saying is after 8 divisions, you would have 256 bacteria in that population. The next thing that we need to cover are stem cells. So stem cells are undifferentiated cells. They have not yet specialized. They can divide to produce many more cells, and these cells will then differentiate or specialize, producing other cell types. Now, there are different types of stem cells. So we've got embryonic stem cells that come from early human embryos. And the benefit of those stem cells is they still have the potential to differentiate into any type of cell, any body cell. So they're really useful in research, and they can be used to treat conditions like paralysis and diabetes. For example, to treat paralysis, they can differentiate into nerve cells, and to treat diabetes, they could differentiate into cells of the pancreas. Another type of stem cell is the adult stem cell. Now, these are found in adults, obviously. For example, you find stem cells in bone marrow and some other tissues and organs. Now, they are restricted in terms of what they can differentiate into. So they're more limited. They can only differentiate into a limited range of cell types. So for example, the adult stem cells that are found in bone marrow can differentiate into a few different cell types, but only blood cells. So red blood cells and white blood cells, for example. They are used in medicine. So you could use them for bone marrow transplants. You could use them to treat leukemia, to replace the damaged blood cells in a patient with leukemia. And the third type are plant stem cells. These are found in what we describe as the meristem regions of plants. Now, meristem regions, where the plant stem cells are located, are found in the growing regions, so such as the tip of the growing shoot and the tip of the growing root. They will contain plant stem cells. And what's interesting about these plant stem cells is they have the potential to differentiate or specialize into any type of plant cell, but they maintain this ability throughout a plant's lifetime, which is why you can take a cutting from a plant, you can cut off a side shoot or a stem, and the stem cells will be able to divide and differentiate into new root cells, for example, so you can clone a plant. Used to produce clones of plants quickly and cheaply, as I've just said, taking cuttings, which comes up on paper 2, would be an example of that. Let's go through some of the uses of stem cells and start to think about the advantages and the disadvantages of the two main types, the embryonic and the adult stem cells. So embryonic stem cells, because they come from an early embryo, they still have the potential to differentiate into any cell type. So they are highly versatile. They divide rapidly and they are easy to grow in the laboratory. Because they can differentiate into many cell types, they have the potential to treat many conditions, as we've said already, including paralysis, diabetes, also heart disease would be another example that you could give. There are some disadvantages with embryonic stem cells. So obviously ethical issues, but do clarify what you mean by that. You are getting these embryonic stem cells from an early embryo. And when you extract these stem cells, that embryo is going to be destroyed. Now, some people view this as destroying a potential life because the embryo had the potential to continue to grow and to develop into a fetus. Destroying the embryos that the stem cells are obtained from does raise ethical concerns. Also, because embryonic stem cells divide rapidly, that rapid cell division could lead to tumour formation and possibly cancer. And you've got the problem of immune rejection. So if you're using embryonic stem cells that do not match the patient's cells, then those transplanted stem cells could be rejected and destroyed by the patient's own immune system. So the transplanted stem cells would not be successful. Let's think about the adult stem cells. Now, you don't have the same ethical concerns when using adult stem cells because they are taken from an adult donor, for example, the stem cells from within their bone marrow, and the adults can obviously give consent to have their stem cells taken. If you are taking stem cells from the same patient, you don't have that risk of rejection. So the transplant would be safe in terms of the cells would not be rejected by the patient's own immune system. It has already been used successfully. So using bone marrow transplants from a donor to treat someone with blood cancer, that has been used successfully. So it's quite a reliable and tested procedure. Disadvantages of adult stem cells. Remember, they can only differentiate into a limited range of cell types, like the bone marrow stem cells can only differentiate into blood cells. So they are more limited in terms of what they can treat. They're also harder to grow and manipulate in the lab. So harder to grow and harder to encourage to differentiate into the cell type that you need. And because these are coming from adult donors, the cells may have accumulated mutations or changes in their DNA. Now, what we can look at is a process called therapeutic cloning. Now, this is a process where an embryo is created which will have the same DNA as the patient that needs treatment. And this is done so you can get the embryo and take the embryonic stem cells, which obviously can differentiate into any cell type, so that would be really useful. But because the embryo produced was a clone of the patient, the embryonic stem cells will also be genetically identical to the patient's cells. So it overcomes that issue of rejection by the patient's immune system. Now, how does this work? Let's go through it step by step. So a nucleus is taken from a patient's body cell. So this is the person who needs the stem cell transplant or needs the new tissue or the new organ, the patient, right? You're going to take an adult cell from that patient and you're going to remove the nucleus, because obviously the nucleus contains all of that patient's DNA. It's found within the nucleus. That nucleus is inserted into a donor egg cell. So we've taken a human egg cell from a donor. We've removed the nucleus from the egg cell so it does not contain the donor's DNA. And you're going to put the nucleus that you got from your patient that has all of the patient's DNA within it into the empty egg cell. Now, that cell is then stimulated and it will start to divide and it will form an embryo, an early embryo. Now, this early embryo contains embryonic stem cells that can differentiate into any cell type, but because you made that embryo using the nucleus from the patient, it has all of the patient's DNA. So the embryo is basically a clone of the patient. It is genetically identical. The stem cells can then be extracted from the early embryo and grown in the lab. And these stem cells can be directed to differentiate into any cell type. So if you need cardiac muscle cells for the heart, or kidney cells, or nerve cells to treat a damaged spinal cord, or maybe cells of the pancreas that can produce insulin to treat a diabetic. Remember, these cells can differentiate into any cell type, so can then potentially be transplanted into the patient to treat any damaged tissues. Now, what are the advantages of this process? Because the embryo created was genetically identical to the patient, there is no risk of rejection by the patient's own immune system. They can be used to treat many different diseases, so make sure you can give some examples: Parkinson's, diabetes. You can make new nerve cells to treat spinal injuries, or cardiac muscle cells to treat heart disease. Remember, they can differentiate into any cell type because they are embryonic stem cells. Disadvantages and ethical concerns. Again, this involves embryo destruction. You are creating an embryo that is genetically identical to the patient, or a clone, to remove those embryonic stem cells. Remember, an embryo is a potential life, so it does raise ethical concerns. Some believe it's unethical just to create an embryo for research or treatment purposes, and this is still very much experimental. So success in treatments is limited. So we don't know if it's going to be successful. We don't have much reliable data to base that information on. Again, just like with other types of stem cells, because the stem cells divide rapidly, when they are put into the patient, there is a risk of tumour development if those stem cells continue to divide uncontrollably. It could lead to tumour development and potentially cancer. The next part of Module 1 is all about transport of substances in and out of cells. So the first transport method we're going to look at is diffusion. So what is diffusion? Diffusion is the net movement or the overall movement of particles from an area of higher concentration to an area of lower concentration. Or because it's going from higher to lower, we can say down a concentration gradient. It is passive, so diffusion does not require energy from respiration. It may take place across a partially permeable membrane, but it doesn't have to. So if I spray some air freshener, the air freshener particles will diffuse from the region of higher concentration where I've just sprayed them across the room to a region of lower concentration. That's diffusion, and that does not involve a partially permeable cell membrane, so you don't have to have one. Obviously, in biology, we're often talking about diffusion in and out of cells, so that usually is a partially permeable membrane in biology. Here we've got an example. So the red represents a partially permeable membrane. And if you look at the diagram on the left-hand side, we've got two different regions. We've got a region of higher concentration on the left-hand side of the membrane and a region of lower concentration on the right-hand side of the membrane. So all we need to remember is that diffusion is the net movement of particles from higher to lower concentration. So these particles are going to diffuse in that direction. And that diffusion will continue until an equilibrium is reached, which means there's equal concentrations on both sides. In that case, there's no concentration gradient anymore, so diffusion would stop. The concentration gradient is just the difference in concentration. So once equilibrium has been reached, we would say there's no longer a difference in concentration, or there's no longer a diffusion gradient, so diffusion would stop. Examples of diffusion in biology that you might see in your exam. So oxygen diffuses into cells for respiration. Carbon dioxide diffuses out of cells because it's made in respiration and it's a waste gas that cells want to get rid of. Glucose can diffuse into the blood from the small intestine. So when we've eaten, we've digested carbohydrates. If we've got a higher concentration of glucose in the small intestine, then it can diffuse down the concentration gradient if there's a lower concentration of glucose in the blood. What factors affect the rate of diffusion? Let's go through the main ones. So the first one would be concentration gradient, which is the difference in concentration between two areas. And the greater the difference in concentration between two areas, the faster the rate of diffusion. So what we say is there's a steeper concentration gradient, the rate of diffusion will be faster. A steeper concentration gradient is just a gradient where there's a big difference. So you've got a very high concentration in one region and a very low concentration in the other region. That would be a steep concentration gradient, and the particles will diffuse more quickly. Now remember, as diffusion continues, the gradient would become shallower or less steep. So diffusion would slow down, and when an equilibrium is reached, where the concentration is even or equal in both regions, then diffusion would eventually stop. The second factor that affects diffusion is temperature. Now this one's quite easy to understand, I think. If you have a higher temperature, it's going to speed up the rate of diffusion. And this is because higher temperatures give particles more kinetic energy, so they're going to move faster. And if they're moving faster, they're going to diffuse faster. It's that simple. The third factor is surface area. So if there is a membrane involved, which there often is in biology, remember there doesn't have to be for diffusion, but there often is in biology. If we've got a cell membrane, or the wall of the alveolus could be the surface area we're talking about, or the lining of the small intestine could be the surface area that we're talking about. The bigger the surface area, the more particles can pass through at the same time. So the bigger the surface area over which the particles are diffusing, the faster the rate of diffusion. And this is why cells adapted for diffusion, or surfaces adapted for diffusion, like the alveoli or the small intestine, will have adaptations to maximize their surface area to increase the rate of diffusion, because the bigger the surface area, the more particles can diffuse at one time. The next thing to think about is the thickness of the exchange surface. So if diffusion is taking place over a surface, over a membrane, over a wall, then the exchange surface needs to be thin. If the exchange surface is thin, for example, only one cell thick, like the walls of the alveolus are only one cell thick, that is a short diffusion pathway. So substances can move across much more quickly. They literally just don't have to go as far. So the shorter the distance, the shorter the diffusion pathway, the faster the rate of diffusion, or the faster the particles can diffuse. And we're going to look at examples of exchange surfaces where diffusion takes place. And we're going to be seeing these features. So we're going to be seeing a thin exchange surface and a large surface area. We're also going to be seeing ways that we try to maintain the concentration gradient. So diffusion always occurs and an equilibrium is never reached. So let's look at our first example. This is your respiratory system. So let's look through it together. Obviously, we've got the nasal cavity, the nose. Then when we breathe in, air goes down our trachea, or our windpipe. It goes down our two bronchi. One goes into the left lung and one goes into the right lung. They then branch into smaller tubes called bronchioles, and bronchioles end in these air sacs called alveoli, and this is where gas exchange takes place. So what are alveoli? They are tiny air sacs in the lungs. You have millions of them, and they are the site of gaseous or gas exchange between the air and your blood. They are found at the end of those small tubes called bronchioles in clusters, and as I said, there are millions of them. So what actually happens with gas exchange? Well, oxygen diffuses from the alveoli into the blood, because you breathe in, the oxygen goes into the air sac or into the alveoli, and the oxygen is going to diffuse into your blood so it can be carried around your body and delivered to your cells for respiration. But at the same time, carbon dioxide, which is in your blood, is going to diffuse from your blood and go into the alveoli to be exhaled or breathed out, so we can get rid of that waste gas. Waste carbon dioxide is made during respiration, but we need to get rid of it. So it's going from the blood by diffusion into the alveoli to be exhaled. Now you can see that on this diagram. So this is a single alveolus, just one alveolus. We've got millions of them in our lungs, and you can see the process of gas exchange. So when we breathe in, air enters the alveoli, and that air will have a higher concentration of oxygen. So the oxygen will diffuse from the alveolus into the blood, where it's picked up by a red blood cell and specifically carried by the hemoglobin. So it can be carried by our bloodstream to our cells for respiration, but it's called gas exchange because at the same time, it's like we're exchanging the oxygen with carbon dioxide, right? So the concentration of carbon dioxide is higher in the blood than in the air in the alveolus. So CO2 is going to be diffusing in the opposite direction. Make sure you know the directions. It's going from the blood into the air sac, and you will then exhale and breathe out that carbon dioxide. Now, both of these gases are moving by diffusion. So oxygen is going from a higher concentration in the alveolus to a lower concentration in the blood. And at the same time, carbon dioxide is going from a higher concentration in the blood and diffusing to a lower concentration in the alveolus. Now, the alveoli are adapted for gas exchange to make sure the speed of diffusion is as fast as it can be so that this process is very efficient. So let's go through some of the adaptations, and you should recognize these because they link to the factors that speed up diffusion. So the alveoli have a large surface area. That would be describing the adaptation. If we explain it, because there are millions of alveoli, it gives a large surface area for diffusion, or you could say diffusion will be faster, so the gas exchange can happen faster simply because there's more surface over which the gases can diffuse at one time. The alveoli also have thin walls. So if we look at our alveolus here, this is the wall of the alveolus, yeah? And the wall of the alveolus is thin. Don't say the alveoli are thin. It's the wall of the alveolus that's thin. And it's the wall of the alveolus that the gas exchange by diffusion is taking place over. Those walls are thin. In fact, they are just 1 cell thick. That is going to reduce the diffusion distance, or we can say there's a short diffusion pathway. So again, faster diffusion, simply because there's less of a distance that those gases have to travel across. They also have a good blood supply. So you'll have noticed that the alveoli are surrounded by capillaries. Capillaries are microscopic blood vessels, so really, really small blood vessels. They also have thin walls that are just 1 cell thick, which again reduces the diffusion distance. But because there's such a good blood supply, it means each alveolus is right next to a capillary, right? Again, reduces the diffusion distance, but it also helps to maintain a steep concentration gradient for oxygen and carbon dioxide. Because within these capillaries, these small blood vessels, the blood is constantly flowing. So as soon as the oxygen diffuses into the blood, that blood is carried away, and it's going to be replaced with blood that has a lower concentration of oxygen. So what that means is there will always be a higher oxygen concentration in the alveolus compared to the concentration of oxygen in the blood in the capillary. So we're maintaining the concentration gradient, so equilibrium is never reached and oxygen will continue to diffuse into the blood. At the same time, because the blood is constantly flowing, it is always bringing in blood with a higher concentration of CO2. So there's always going to be a higher concentration of carbon dioxide in the blood, in the capillary, than in the alveolus, which means that carbon dioxide will continue to diffuse out of the blood and into the alveoli. What we say in the exam is the fact that there is a good blood supply maintains the concentration gradient for oxygen and carbon dioxide. So equilibrium is never reached. Diffusion is always going to take place. The other thing that helps with this is continuous ventilation. We are always breathing. And again, we say it maintains the concentration gradient for oxygen and for carbon dioxide, because we are always breathing in air with a higher concentration of oxygen, and we are always breathing out air with a higher concentration of CO2. So we're making sure there is always a concentration gradient for oxygen, so it will keep diffusing into the blood, and there is always a concentration gradient for carbon dioxide, so it will keep diffusing out of the blood. And on top of that, we've got the thin wall of the capillary, the thin wall of the alveolus, short diffusion distance, and a large surface area. This is all to maximize the rate of diffusion and maximize the efficiency of gas exchange. Let's think about another example, gas exchange in fish gills. Now, the gills are the gas exchange organ in fish, and obviously fish live in water, so they are getting their oxygen from the water via their gills. Now, the gills are located on either side of the fish's head. Okay, so you can see a set here. They are located behind a gill cover, which is like a bony flap. It's called the operculum. You don't have to learn that, but it's like a bony flap that covers the gills, and they're on both sides of the fish's head. They're made of gill filaments, which you can see here. Okay, so these are the gill filaments. So you can see already large surface area. Now, if we zoom in or magnify one of those filaments, on the gill filaments, you have these folds. And these folds are called lamellae. So you've got a huge surface area. You've got gill filaments, and the gill filaments are covered in extra folds called lamellae. So collectively, you've got a very large surface area. Now, what's going to happen is oxygen in the water is going to diffuse into the blood, and you can see that the gill filaments are covered in microscopic blood vessels or capillaries. So again, we've got a very good blood supply. The oxygen is going to diffuse from the water into the blood, from a higher concentration in the water to a lower concentration in the blood. And at the same time, carbon dioxide is going to diffuse out of the blood, where there's a higher concentration, and into the water, where there's a lower concentration. Because remember, diffusion is always the net movement of particles from higher to lower concentration. So let's do what we did with the alveoli, and let's think about how the fish gills are adapted to maximize the rate of diffusion and therefore maximize the efficiency of gas exchange. We've already said they have a large surface area. This is because the gills have many gill filaments and many lamellae, so you're going to get more diffusion taking place at one time, or faster diffusion. Just like the alveoli, we've got another example of a thin exchange surface. The filaments and the lamellae are very thin. So again, we've got a short diffusion distance between the water and the blood vessels. So the oxygen and the carbon dioxide do not have to travel far. It's a short diffusion distance, which speeds up the rate of diffusion. They've got a rich blood supply, or you can say a good blood supply, as you can see on this diagram here. This filament, where you can see the folds, the lamellae, they are full of many, many capillaries, microscopic blood vessels. Now, because they carry the oxygen away, there will always be a concentration gradient. It's just like the blood vessels in your alveoli. As soon as the oxygen diffuses into the blood, the oxygen is carried away and it's replaced by blood with a lower oxygen concentration, which means you will not reach equilibrium and oxygen will continue to diffuse from the water into the blood. But what's interesting about the fish gill setup is we've also got countercurrent flow. So the water and the blood flow in opposite directions, okay? So as the fish is swimming in the water's coming in through the mouth and it goes in that direction out over the gills. The blood in the capillaries is flowing in the opposite direction to the water. Now, this is something that you'll cover more at A level, but for GCSE, it's worth remembering it maintains a steep concentration gradient. It will mean that the water will always have a higher concentration of oxygen than the blood it is next to. So diffusion will continue to take place from the water. Oxygen will continue to diffuse into the blood because you will always have a steep concentration gradient. It will always be higher in the water than the blood. Again, we can say equilibrium will never be reached, which means diffusion will continue to take place. Another example of gas exchange where we see diffusion is in the leaf. So what gases are exchanged? Now, obviously when it's sunny, when there's light available, plants are photosynthesizing. And so they need a lot of carbon dioxide. That's one of the reactants for photosynthesis, right? So carbon dioxide is going to diffuse or enter the leaf for photosynthesis. And photosynthesis makes oxygen. So oxygen is going to diffuse out. Now, some oxygen that it makes in photosynthesis may be used by the cells for respiration. But if there's a high rate of photosynthesis and there's a lot of oxygen being made, it might be that some of it gets used in respiration, but the rest of it diffuses out. So this is really when it is sunny, when there's a higher rate of photosynthesis than respiration. You've got CO2 diffusing in for photosynthesis and oxygen diffusing out. Now, where does this happen? These gases diffuse in and out of leaves through stomata. These are tiny pores or little holes that are mainly found on the lower surface of a leaf. And the stomata are surrounded by guard cells. Guard cells can control the opening of stomata to regulate gas exchange and water loss. Obviously, stomata need to be open to allow gas exchange to take place. For example, to allow the carbon dioxide to diffuse in for photosynthesis, to allow the oxygen to diffuse out. But water can evaporate and be lost through those stomata. So if too much water is being lost via those stomata, the guard cells can close the stomata to reduce water loss via evaporation. Now, let's talk about the spongy mesophyll layer. I'm going to show you a diagram of this as well in a second. But the spongy mesophyll layer inside the leaf, it has air spaces. Now, the fact that it has air spaces does mean that gases can quickly diffuse. So they enter via the stomata and they can quickly diffuse through the air spaces in the spongy mesophyll layer and up to the palisade mesophyll cells. Now, remember, we looked at those as a specialized cell. They carry out photosynthesis, lots of photosynthesis. They need lots of carbon dioxide. So it's diffusing in through the open stomata. It's quickly able to diffuse through the air spaces in the spongy mesophyll layer, quickly diffusing up to the palisade cells so they can use that carbon dioxide for photosynthesis. Now let's have a look at this on a diagram. So we can see the stomata here. So this is a little pore or a little gap, usually found on the underside of the leaf, the lower surface of the leaf. Now, it will allow carbon dioxide to diffuse in for photosynthesis, and oxygen that is made during photosynthesis can diffuse out. So they are the site of gas exchange. But remember, water can also be lost through the stomata. It can evaporate and diffuse out of those stomata, which is why the guard cells might close the stomata to reduce that water loss. But let's go back to the gases. So the carbon dioxide, which is needed for photosynthesis, it diffuses in through the stomata and it can quickly diffuse through the spongy mesophyll layer, which is this layer here, because there's lots of air spaces, lots of gaps. So it can quickly diffuse through the air spaces and it can quickly reach the palisade cells, because that's the main site of photosynthesis. They need carbon dioxide, lots of carbon dioxide as a reactant for photosynthesis. Equally, they make oxygen during photosynthesis. The oxygen can diffuse through the air spaces in the spongy mesophyll layer and out of the leaf through the stomata. Now, there are some other adaptations of the leaf, just general adaptations that help speed up the rate of diffusion. So you'll notice that all leaves are thin. I mean, it looks like there's quite a few different tissue layers here, but even with those tissue layers, the leaf is still very, very thin. So that gives us a short diffusion distance or a short diffusion pathway. So obviously diffusion will be faster. The leaf has stomata. That is what is allowing the gases to enter and leave. It's allowing the gases to diffuse in and out. That is the site of gas exchange. We've mentioned the air spaces in the spongy mesophyll layer already. They allow the gases to diffuse quickly between the cells. So for example, up through the spongy mesophyll layer to the palisade cells. And finally, leaves have a large surface area. Most leaves do. They tend to be quite wide and flat to maximize the area for diffusion. Large surface area, faster diffusion. For example, if you've got a large surface area, you're going to have a larger bottom surface with more stomata, more gas exchange. But also having a large surface area does increase light absorption. So we need to mention that as well. Light is needed for photosynthesis, and the wider, flatter, bigger the surface area of the leaf, the more light can be absorbed by chlorophyll to increase the rate of photosynthesis and increase the production of glucose. Let's talk about the small intestine next. I think this is my favorite one. So absorption happens in the small intestine. Now, absorption is the movement of digested food molecules such as glucose from carbohydrates or amino acids from your proteins from the small intestine into the blood. So we're absorbing these nutrients, these digested food molecules from the inside of our small intestine into our bloodstream. They can then be transported in our bloodstream around our body, delivered to our cells. So our cells can use glucose for respiration, to release energy, or use amino acids to make protein so we can grow and repair tissues, etc. Now, where does this absorption happen? It's obviously in the small intestine. Now, the small intestine is very long, which does increase the surface area and the time over which absorption can take place. But really important when we're learning about the small intestine, the lining of the small intestine is covered in projections or folds called villi, and each villus also has a folded membrane. We call those folds microvilli, and the villi and the microvilli massively increase the surface area for absorption. So let me show you this. So here is our small intestine, which is basically like a big tube, isn't it? A big tube that folds round like this in our abdomen. So you can see a portion of it here. It's surrounded by layers of muscle, so it can help to move and churn the food. But if we look inside the small intestine and we look at the inner lining or the inner wall of the small intestine, you can see it here. It's folded, and the big folds are called villi. But on the villi, if you look at the outer layer of cells, which we could call the epithelial cells, the surface cells, on those cells, the membrane is folded. And we call those folds microvilli. So you've got the villi, which are folds, but the outer layer of cells also have a folded membrane. So you've got folds on folds. Microvilli on the villi. You've got a huge surface area over which absorption can take place. And some of that absorption will happen by diffusion, but it does depend on the concentration gradient. So if you've got a very high concentration, let's just draw some imaginary glucose. If you've got a very high concentration of glucose inside your small intestine and a lower concentration of glucose inside your bloodstream, it can diffuse over the wall of the villus and enter the bloodstream, because you'll notice there are capillaries, microscopic blood vessels in these villi. Equally, if you had a very high concentration of amino acids in your small intestine, they could diffuse from a higher concentration to a lower concentration from inside your small intestine into your bloodstream. If the concentration is lower in the small intestine, we do use active transport as well, which we're going to talk about towards the end of this presentation. But let's go ahead and go through the key adaptations of the small intestine, because this could come up as a 4 to 6 marker. So we've already said inside the small intestine there are villi, the big folds, and microvilli, which is the folded cell membrane. This increases the surface area. We've seen this a lot now. The bigger the surface area, the faster the rate of diffusion, or in this case, the faster the nutrients will be absorbed into the bloodstream. Now, the walls of the villi are only 1 cell thick. So if you look at the diagram here, I can see on this villus, if you look at the wall specifically, this bit here, the wall of the villus is just 1 cell thick. Again, don't say the villi is 1 cell thick. Same way we don't say the alveoli is 1 cell thick. We say the wall of the alveolus is 1 cell thick, or the wall of the villi is 1 cell thick. So this gives a short diffusion pathway, because the nutrients don't really have to diffuse very far at all to reach the bloodstream or to reach a capillary. Each villus contains many capillaries, or you can say there's a good blood supply, which again maintains a steep concentration gradient, because the blood is constantly flowing and carrying those nutrients away. The small intestine has walls made of muscle. This keeps the food moving as the muscle contracts. So it keeps churning and it's moving the nutrients, trying to move them closer to the walls of the small intestine, or the walls of the villi, so the nutrients can be absorbed. Many mitochondria in the surface cells, or the epithelial cells. So the surface cells are the ones that I put a little box around before. These surface cells here, yeah? We've magnified one of those surface cells over here. It's called an epithelial cell. It just means surface cell. But these surface cells that form the wall of the villi do contain many mitochondria. Now, we haven't covered active transport yet in this video. It's coming up shortly. But sometimes nutrients do need to be absorbed by active transport. If there's a lower concentration in the small intestine than in the blood, we're going to have to use active transport. So that requires energy from respiration. Makes sense as to why we'd have many mitochondria. Mitochondria is the site of respiration, so it will release or provide energy for the active transport of those nutrients. And finally, a nice easy one to remember if you want to, the small intestine is very long. The food spends quite a long time traveling. I think it's about 7 meters in total. So that just gives more time for absorption to take place. Let's think about plant roots, another example of exchange. Now, plant roots, they have various jobs. They anchor the plant in the soil. They hold the plant in place. They absorb water from the soil and minerals that they need for growth. So plants need water for photosynthesis. It's one of the reactants in photosynthesis. They also need it for transpiration, so it will move up the xylem in the transpiration stream and it will evaporate out of the stomata. Helps to keep the plants cool. Also does help to maintain the shape of cells, because if a cell is full of water, we say it's turgid. We're going to do that in a second, but it helps to strengthen and support the cells and give the plant overall structure. So water is very important. Mineral ions, which plants get from the soil, also very important. They need nitrate ions to make amino acids and proteins, for example. They need magnesium as another example of an ion. They need magnesium to make chlorophyll. They also need phosphate ions and potassium ions. But there's just a couple of important examples for you there. So how are these substances absorbed? Well, it's not quite diffusion. Water is actually moved into the roots by osmosis. But osmosis is diffusion. It's just diffusion of water. Minerals are absorbed usually by active transport against the concentration gradient, because there's usually a lower concentration of mineral ions in the soil than in the plant's root cells. Now, active transport does require energy from respiration to move those minerals against their concentration gradient. We're going to be covering osmosis and active transport in a second, so hang fire. So how are roots adapted? We're going to see loads of the same features. They have root hair cells. We've seen them before. We looked at them as an example of a specialized cell. They have this big projection that we call a root hair. And that long extension increases the surface area, which is going to maximize the amount of water and the number of mineral ions that can be absorbed. Okay, so this kind of links to this. The root hair is what provides the large surface area, but also there are just many roots, right? They're highly branched, they go really wide, they go really deep. So not only have you got root hair cells that increase the surface area, but the whole root network just has a really large surface area to maximize absorption. They do have a cell wall because they're a plant cell, but it's a thin cell wall, so you've got a short diffusion distance. Remember, root hair cells don't have chloroplasts. They're not needed because it's dark underground. There's no light for photosynthesis. So it saves the cells energy. They don't have to make those chloroplasts. Importantly, root hair cells have many mitochondria because when they do actively transport mineral ions, they need energy for that, which comes from respiration, which takes place in the mitochondria. And there will be a steep concentration gradient maintained because as soon as the water and the mineral ions move into the roots, they will be moved into the xylem, and the xylem will transport them up the plant, away from the roots, up the plant in the stem. So you're trying to maintain the concentration gradient so water keeps diffusing in by osmosis. Remember, it's very difficult with mineral ions, so we are usually having to use active transport because they often are having to go from low to high. But at least with water, if it keeps moving away and up the xylem, we're going to have a higher concentration of water in the soil than in the root hair cells, so the water will keep moving in by osmosis. Okay, let's think about osmosis next. We've just mentioned it with root hair cells. Osmosis is the diffusion of water across a partially permeable membrane. So this has to take place across a partially permeable membrane, which in living things will be the cell membrane. And the water will move from a dilute solution to a concentrated solution. Now, this confuses students very often. A dilute solution will have a higher concentration of water, but a lower concentration of solute dissolved in it. So we always say that osmosis is the diffusion of water from a dilute solution because a dilute solution has a higher concentration of water. Just think about dilute orange squash. It has less orange squash, it has more water, right? So it's going from a dilute solution to a concentrated solution, but a dilute solution does have a higher concentration of water. So it's the same as saying water is diffusing from a higher concentration of water to a lower concentration of water, or from a dilute solution to a concentrated solution. Just really simple. It's going to go from where there's more water to where there's less water, okay? But try to use these words in the exam, from a dilute solution to a concentrated solution. Now, osmosis is a passive process because it is a type of diffusion, and diffusion is passive, so it does not require energy from respiration. The cell membrane is going to be our partially permeable membrane. Now, it's partially permeable. It only lets some substances through and not others, but water can pass through the cell membrane. However, it can control the entry of sugar or salts. Osmosis is not the movement of sugar or salt. Osmosis is simply the diffusion of water from a dilute to a concentrated solution. So if we look at this kind of experiment here, we've got a partially permeable membrane in the middle. So we've got two different regions. Now, if you look over on the left-hand side, this is a dilute solution because you've got a higher concentration of water, and the little pink blobs, they are the solute, they are the sugar molecules. So on the left-hand side, there's less solute, there's less sugar, and there's a higher concentration of water. So this side is our dilute solution. If you look at the right-hand side is concentrated because it's got more solute or more sugar molecules dissolved in the water. It's got a higher concentration of solute, lower concentration of water. So osmosis is going to occur in this direction because remember, osmosis is the diffusion of water from a dilute to a concentrated solution. Or we can say it's the diffusion of water from a higher concentration of water, which is a dilute solution, to a lower concentration of water, which is a more concentrated solution. Where does osmosis happen? So we've looked at root hair cells already. Water moves from the soil into the root hair cells by osmosis from a dilute solution in the soil to a more concentrated solution in the root hair cells. Happens in animal cells. Root hair cells are plant cells, right? But it also happens in animal cells, like red blood cells are quite often used in the exam. Animal cells can gain or lose water by osmosis, just depending on the concentration of the surrounding solution. Plant cells in general, water again can move in or out of the cytoplasm, in or out of the vacuole, which also contains water, and it will affect the pressure inside the plant cell. So what we're going to do next is look at specific examples of osmosis in plant cells and animal cells, because plant cells have a cell wall, so they do behave differently when water enters the cell. So let's have a look at how osmosis can affect plant cells. So if you put plant cells into different concentrations of solution, they would either gain water and gain mass or lose water and lose mass as a result of water moving in or out by osmosis. So if you put a plant cell in pure water, this is the most dilute solution, right? It's just water with no solutes. So water would enter the plant cell by osmosis from a dilute solution outside the cell to a higher concentration inside the cell. If water enters, the cell would gain mass. And we say plant cells become turgid because the cytoplasm is full of water, the vacuole is full of water, and they are pushing out against the cell membrane. But remember, plant cells have a cell wall, so the cell wall is strong enough to withstand that pressure. So that's why we use the word turgid to describe the plant cell. If you place a plant cell in a solution that has the same concentration as inside of the cell, this would be described as an isotonic solution where the concentrations are equal. There'd be no net movement of water by osmosis. I mean, it would be moving in and out, but equally. So no overall or no net movement of water by osmosis. So no change in mass of the cell. If you place a plant cell in a concentrated solution, so a solution with like a high concentration of sugar or salt, water is going to leave the cell because there's a more dilute solution inside the cell than outside of the cell. So the water moves by osmosis from the dilute inside the cell to the solution outside. If water leaves by osmosis, the cell will lose mass. The cell will become flaccid, where the cytoplasm and the vacuole start to shrink. It could even become plasmolysed, where the plant cell has lost so much water that the cell membrane actually starts to shrink inwards and pull away from the cell wall. Let's have a look at these in diagram form. So the cell here has been placed in pure water. So water is going to move in to the cell. The cell is going to gain mass and become turgid. Here we've placed the cell in an isotonic solution, a solution that has the same concentration as inside the plant cell. So you get movement in and out, but there's no net movement of water by osmosis because the concentrations are equal. So the shape and the size and the mass of the cell stays the same. Here you've got a plant cell that has been placed in a concentrated solution. So outside of the cell, there's a high concentration of salt, for example. Water is going to move out by osmosis. So you can see here, the cytoplasm has started to shrink. The cell membrane has started to pull away from the cell wall. So the cell becomes flaccid, and then even more extreme, once the cell membrane pulls away from the cell wall, we say the plant cell is plasmolysed. But these terms, turgid, flaccid, plasmolysed, we only use them for plant cells. Plant cells are unique because remember they have a cell wall made of cellulose and it's strong, so they can withstand the pressure exerted when water enters the cell. So in this first example here, if you place a plant cell in a very dilute solution like pure water, water moves in. We say it becomes turgid because the cell wall is strong enough to withstand that pressure. So the plant cell would not burst, it would become turgid. And that would actually help to support the plant. If the cells are turgid, it gives the plant strength and support so it can stand upright and it won't wilt. Now, this is not the same as an animal cell. Animal cells don't have cell walls, so if too much water enters an animal cell by osmosis, the cell would burst. Let's look at animal cells now. So this is different, right? Remember, they don't have a cell wall. So if you place an animal cell, we're going to look at red blood cells as the example. If you place one in pure water, which is dilute, water's going to move into the animal cell by osmosis. The cell would gain mass, and because it doesn't have a cell wall, there's no cell wall to withstand that pressure, so the cell may burst, which we call cell lysis. If you put an animal cell into a solution that's the same concentration, same as the plant cell, there'll be no net movement of water, no change in mass of the cell, so the size, the shape, the mass would stay the same. If you put an animal cell into a concentrated solution, a solution that has a high concentration of solute, like salt or sugar, water would leave the cell because inside the cell is more dilute and outside is more concentrated. So water would leave by osmosis, the cell would lose mass, it would shrink or shrivel, which we say is crenation. So the cell crenates. Let's have a look at some red blood cells. So here you can see cell lysis. So this is a red blood cell that's been placed in pure water. So lots of water has moved in from a dilute solution outside the cell to a concentrated solution in the red blood cell. So much water's moved in, there's no cell wall to withstand the pressure here, so we've got cell lysis. The cell has burst. Here you've got a red blood cell in an isotonic solution, same concentration outside the cell as inside. So you can see the size, the shape, the mass of the red blood cell stays the same because there's no net movement of water by osmosis. And here you can see a cell that has undergone crenation. So much water has left the red blood cell because inside the red blood cell was more dilute, and outside you've put it in a solution that's very concentrated. So water's left by osmosis and you can see the cell has started to shrink or shrivel or crenation has taken place. Now, something else we have to think about with osmosis, just like diffusion, is key factors that affect the rate of osmosis or how quickly that water will move by osmosis across the partially permeable membrane. So concentration gradient, the steeper the concentration gradient or the steeper the difference in water concentration, the faster the rate of osmosis. So if you've got your cell in a solution of pure water and inside the cell it is very concentrated, water would very quickly move in from the dilute solution outside to the concentrated solution inside. So the steeper the concentration gradient, just like diffusion, the faster the rate of osmosis. Temperature will affect it as well because it is just the movement of water particles. Remember, osmosis is just diffusion of water. Higher temperatures mean that water molecules would have more kinetic energy, they'd be moving faster, so you're going to get faster osmosis from that dilute to that concentrated solution. Remember, osmosis has to take place across a partially permeable membrane, so the surface area of that membrane is always important, and the bigger the surface area, the faster the rate of osmosis, simply because there's more surface over which the molecules of water can move at one time. The thickness of the exchange surface, again, is important because the water molecules are crossing over that exchange surface. So if the exchange surface is thinner, that is a shorter diffusion pathway or a shorter osmosis pathway. So water can cross more quickly. You're going to get faster osmosis if you've got a thinner surface over which osmosis is taking place. Now, there is a required practical linked to osmosis, so let's have a look at that. The aim of this practical is to investigate the effect of different concentrations of sugar solution on the mass of plant tissue due to osmosis. So you probably use potatoes as the plant tissue, and you can identify the concentration of sugar inside the plant cells. Now, you're going to use potatoes when you do this in school, but in the exam, I've seen them with questions on carrots and eggs that have had the shell removed, but it's the same knowledge that you're going to apply, just to a different cell type. Let's go through the method. So you would be cutting equal-sized cylinders of potato. So you use a cylindrical cork borer and a ruler to make sure they are the same size and hopefully the same surface area. That is a controlled variable. You measure the initial mass, so the starting mass of each cylinder using a balance, and you record it. You would then place each cylinder or each piece of potato into a test tube that has a different concentration of sugar solution. So you're going to have a range of different concentrations. We're going all the way from pure water, which has a concentration of 0, up to a very concentrated sugar solution that has a concentration of 1 mole per decimeter cubed. And you can see we've gone up in increments of 0.2. So we've got a range of different concentrations. We're increasing the sugar concentration as we go from left to right. The solution should be the same volume. The potato cylinders should be the same size, same surface area. They should have had their skin removed because that would act as a barrier to osmosis. And obviously you're going to leave them in there for the same amount of time. And I've put here at least 30 minutes because you need to allow enough time to actually record a change in mass. You need to give time for osmosis to take place. If you left them longer, you'd see more of a change in mass, but if you leave them too long, some of the potatoes will just turn to mush. So 30 minutes is a sensible time. An hour is a sensible time. I'd probably go with an hour, but you should be leaving them all for the same amount of time. Otherwise, you're not controlling that variable and you will get more time for osmosis in some than others. Once you've left them for that same amount of time, you're going to remove the cylinders from the solution and you're going to blot them gently with a paper towel to remove any solution that may be stuck to the surface, because that would increase the mass, wouldn't it? And you would not get accurate results. So you blot them with a paper towel, then you record the final mass. You've then got your mass before you put them in the solution and your mass after you'd left them in solution for your set time. So you could then work out the difference in mass, but what we suggest you do is actually calculate the percentage change in mass so that you can actually compare your potato cylinders. Even if the starting masses were slightly different, if you work out the percentage change in mass, it makes them all comparable. So to do percentage change, it's the difference over the original times 100. So to find out the difference, you subtract the starting mass from the final mass. That will give you the change in mass, the difference, over the original, which in this case is obviously the starting mass. And then you multiply that by 100 to get it as a percentage. Now, in our investigation here, the independent variable, so the one that we changed, was the concentration of the sugar or the sucrose solution. And we had 6 different concentrations. We're going to get a nice range of results, which is enough to plot a graph. The dependent variable is the one you measure, the one that you're recording in the table. So it's going to be the change in mass that you work out, or you're going to hopefully do the percentage change in mass so that you can compare all your cylinders, even if they did start out at slightly different masses. Now, to ensure that the experiment is fair and the results are valid, All variables except the concentration of the sugar solution must be controlled. And it is really important to include any control variables if you are asked to plan an investigation in an exam. We want to be certain, don't we, that it is the concentration of the solution that is causing those differences in the change in mass and not something else that we have forgotten to control, like one of them still had potato skin on or one of the cylinders was much bigger. So a list here of some of the key control variables: the size and the shape of the potato, so the length, the diameter of the potato cylinders. If you use the same cork borer that has the same diameter and you make sure your potato cylinders are the same length by measuring them with a ruler, then the size and shape should be the same, and hopefully the surface area to volume ratio has been controlled. Use the same volume of solution in each test tube, and you should be using enough so it fully covers or submerges your potato cylinders. Time in the solution is really important, so leave them all in for the same time period. 30 minutes or an hour would be sensible. Temperature. We know temperature affects the rate of osmosis because it would affect how much kinetic energy the water molecules have. So you could do the experiment at room temperature, so they're all at the same temperature, or you could do this in a water bath set at 30 degrees Celsius. If your water bath is set at a slightly warmer temperature than room temperature, it will speed up the rate of osmosis, which means you might be able to leave them for a shorter time, for example, 30 minutes, which means you could probably do this during a lesson in your school. If it's too cold and osmosis takes too long, it may take you longer than a lesson and you wouldn't be able to do the practical during your science lesson. Surface drying. We said you remove the excess solution from the surface by blotting the cylinders with a paper towel. If you did not do this, it would introduce an error. You'd have some solution stuck to the outside of your potato cylinders and it would artificially increase your final mass measurement. But you should dry it in the same way. You should blot them all with a paper towel for the same amount of time, or the same number of blots, or the same number of rolls on the paper towel. Don't blot one more than the other, otherwise you're not controlling that variable either. Same potato source, so same type or same species of potato. Maris Piper potatoes, that's the only one that I know, to reduce variation in plant tissue, because different potato species, different differences in genetics, they may respond differently. So use the same type of potato, or ideally you could get all of your cylinders from the same potato if you use that potato efficiently and you manage to cut all your cylinders from the same one. Now, other things to think about. We should repeat the experiment with any experiment in science. We say repeat at least 3 times at each different concentration. That means you can calculate a mean increase in mass or decrease in mass and a mean percentage change in mass for each concentration. If you do get any anomalies, so results that do not fit your pattern, they do not fit with the rest of your results, those results should be discarded and not included in your mean calculation. And we do recommend calculating percentage change in mass to allow you to compare the potatoes, even if they had different starting masses. Right, when you've got your results, you can plot a graph that looks like this. So on the y-axis, the y1 goes up high, y rhymes with high. We're going to plot the percentage change in mass. And on the x-axis, x is across, we're going to plot the concentration of sugar solution. So we had concentrations going from 0, 0.2, 0.4, all the way to 1, I think. Now you'll notice that our axes look like a T shape. That's because some of our percentage change in mass, they were percentage increases because they gained water by osmosis and they gained mass. But some of our percentage change in masses are percentage decreases because some of our potatoes, if they were in very concentrated solutions, they will have lost water by osmosis across the partially permeable membrane and they would have lost mass. Now the point where the curve crosses 0, that is how we figure out the concentration inside the potato cells, because this point here is telling us there was 0% change in mass. Yeah? Where our line of best fit crosses the x-axis, that represents 0% change in mass. If there was no change in mass, that tells us there's been no net movement of water by osmosis. It has not gained mass, it has not lost mass, there's been no net movement of water by osmosis. So that concentration must be equal to the concentration of solution inside the potato cells. Let me see if I can explain that clearly. It says we know this because there was obviously no net gain or loss of mass. Therefore, at this concentration of solution, there is no net movement of water by osmosis. So this indicates the concentration of that sugar solution must have been equal to the concentration inside the potato cells. That's what we call an isotonic solution. Now, in this graph, the line of best fit has been drawn nicely. It's a smooth curve. It goes through the center of all the points. The concentration where it crosses the x-axis is 0.27, I would say. It's hard to say without all the grid lines on the graph, but it's about 0.27. So what we can conclude from that is the concentration inside the potato cells must be 0.27, because in a solution that had a concentration of 0.27, there was no percentage change in mass, therefore no net movement of water by osmosis. If we just look at one other result, this one here, this potato cylinder was placed in the strongest concentration of sucrose solution, 1 mole per decimeter cubed, and you can see it's had a significant percentage decrease in mass. Now that should make sense to you guys now. That's because it has been losing water by osmosis, because you've placed those potato cells in a very concentrated solution. The solution inside the potato cells was more dilute, so the water moved out of the cells across the partially permeable membrane. Because water's moving out, it had a significant decrease in mass. The last mode of transport to look at is active transport. Now, active transport, different to diffusion, different to osmosis. Active transport is the movement of substances against a concentration gradient. What we mean by that is substances are going to move from a lower concentration to a higher concentration. Now, because the substance is moving against the concentration gradient, this does require energy from respiration. It is not passive like diffusion and like osmosis. So where does active transport happen? In plants, you're going to look at root hair cells. They absorb mineral ions such as nitrates or magnesium ions from the soil, even when their concentration is lower in the soil than the root cells. Those mineral ions can still move in by active transport, which does require energy. And this explains why root hair cells have many mitochondria to release that energy for that active transport. It also happens in animals. So in the small intestine, we spoke about how glucose and amino acids can diffuse from the small intestine into the blood. But what if there's a lower concentration of glucose and amino acids in the small intestine than in the blood? In this case, we need to absorb them against their concentration gradient from a lower concentration to a higher concentration. That's when we would use active transport, which again requires energy from respiration. And this is why the cells that line your small intestine, the ones with the microvilli, also have many mitochondria to release energy for that active transport. And this makes sure that all the nutrients are absorbed into the blood. No nutrients are wasted. We absorb all of the glucose and amino acids, even when there's only a low concentration left in the small intestine. Now, the key thing to remember is that active transport does require energy. So anything that affects energy supply or limits energy supply will limit its rate. It requires energy from aerobic respiration. So aerobic respiration takes place in mitochondria. If a cell has more mitochondria, like a root hair cell, it can respire more, it can release more energy that would increase the rate of active transport. If there's anything inhibiting respiration and stopping the release of energy, then the energy for active transport will not be available, and that would reduce the rate of active transport. Equally, think about this. If cells are in low oxygen environments, so for example, root hair cells that are in poorly ventilated soils that have very little oxygen, with less oxygen, that would reduce the rate of respiration, which means less energy would be released, and that would slow down the rate of active transport. I hope you enjoyed the video and found it useful. If you did, make sure to hit the subscribe button because we are going to be filming more videos like this covering everything from GCSE biology, and they will be posted very soon, so you won't want to miss out. Another reminder about my resources. Remember, these study notes are free. The link is in the description. They have entire topics covered as well as full explainer videos, key terms and extra practice questions. If you're interested in signing up for one of the biology courses here at My Headspace, again, just click the link in the description. The courses are taught by us teachers here at My Headspace. You have 2 live lessons every single week, as well as professionally designed workbooks, exam-style homeworks with video solutions. Basically, everything you need to boost your GCSE grades. I'll see you next time for the next video.